Hello, students. This is Dr. Faizan Mirza. In this video, I'm discussing with you the questions related to genetic technology, how the restriction endonucleases are actually working, what are the restriction sites, and where are they producing the cut, and how gel electrophoresis can be used for identifying these fragments. Okay, so let me share my screen with. Okay, so here this question actually deals with this question came in Feb March 2018 variant 42, and this question deals with how the HbA and HbS carriers can still have healthy kids. So they are saying beta globin polypeptide hemoglobin 2 alleles HbA and HbS. We know this very well. There are three possible phenotypes. We know these. This is sickle cell anemia. This is sickle cell trait, and this individual is normal. A men and women who are carriers, they can choose to have children by IVF. The genotype of embryos can be determined by gene testing before the embryos are implanted. So this removes the idea of, for example, if their baby was having HBS and they had to go for prenatal testing and they by amniocentesis chorionovular sampling, they would have to go for abortion. So that this removes that idea because the embryo is being checked before implantation. So the only healthy embryos will be implanted. Embryos with the normal genotype can then be selected and implanted into the mother. One technique to test the gene in the embryo of HbS allele is restriction fragment length polymorphism. Now, RFLP analysis is a new technique which is shown to you, although it's a very common technique. This in verbose is not there, this is not in the syllabus. So this information is already given to you in the question. So we just need to follow the information and solve the question accordingly. This involves digesting the DNA sample on from an embryo with a restriction endonuclease and then separating the DNA fragments on gel electrophoresis. So you are using restriction endonuclease first, and then you are applying gel electrophoresis. The position of DNA fragments on gel can show if the embryo is having HbS allele. The first step in testing HbS allele by RFLP is using requires having many copies of the beta globin gene, which is where the sickle cell anemia may occur. Name the technique which is used to produce many copies of the DNA. So that's PCR or polymerase chain reaction. Explain why it is necessary to copy this DNA sequence many times in order to test the embryo for HbS allele by RFLP analysis. We know that when the DNA is collected from the embryo, it will be in a very small quantity. So it must be amplified enough to make it visible on the gel when we are using gel electrophoresis. So the next they are saying that in the next step of RFLP, the copies of the beta globin of gene was basically incubated with the enzyme NOST2. The enzyme cuts a specific DNA sequence at the restriction site. The restriction site is shown to you. This is the restriction site. So you need to understand the restriction site of this must have CCT at 5' end, then N. N can be any nucleotide of these, and AAG toward the 3' end. So wherever you have this situation, it will produce a cut. So you can see this is the same condition, this is the same condition, this is the same condition. So you can see that these are the points where this can cut. So you can see that there are many spots which you can see here, which basically this can produce a cut at. So we can this was just my some working here which I was doing, how the fragments and the base pairs are there. So I'll just probably not go into detail of that because that's not very relevant. So we just need to see where this situation is, where this nucleotide sequence is located. So it's located here, it's located here, it's located here. But if we compare the HB, this is HBA allele. Now, if we just compare the HBS allele with it, so HBS allele is having the same sequence at this site as well as at this site, but here the sequence is changed. So there's a single base pair substitution here, and this single base pair substitution changes the restriction site. So this restriction site will not be cut by this enzyme anymore. So when this doesn't get cut by the enzyme anymore, so obviously this means that now this is the change sequence. So this restriction enzyme cannot cut this HBS at this point. So it will cut HB at this site, at HBS at this site, and this site will not be cut. With reference to figure 3.1, 3.2, and 3.3, explained by the enzyme MST2 can be used in the RFALP analysis to show differences between these parts of HB and HBS. We know the allele of HBS is having a point mutation. This is the registration site and MST2. This allele cannot be cut and this site anymore. So HBS allele ends up having two restriction site, and this breaks it into 3 fragments as compared to 3 restriction sites and 4 fragments for HB allele. The size of the fragments would differ as well. HBS allele would have a larger fragment which would travel shorter distance on the gel. We know this for the fact that larger fragments travel shorter distance on the gel. After cutting this with the DNA, they were separated by gel electrophoresis. Explain how gel electrophoresis separates DNA fragments cut with the restriction nucleus. The DNA fragments are poured each into separate wells, and the cathode is attached. We know the buffer solution is added to maintain the pH. DNA being actively charged, they start because of the phosphate group, they start to move to anode when the current is applied. Larger fragments, they travel shorter distance on the gel, so this information can be added. Now you can see the gel is shown to you. They are saying 4 embryos, 1, 2, 3, and 4 were tested for HBsLE using RFLP analysis. 3.4 shows the DNA fragments separated by the gel electrophoresis for the 4 embryos. The DNA fragments of our 2 individuals of known genotype and homozygous A for HBA and homozygous for HBS are shown. So this individual and this individual are the standards here. You can see that if the individual is having HBA, so these are the points where the fragments will have the positions on, and if the HBS is present, so these are the points where the position of the band will look like. So these are our standard, and these are 1, 2, 3 for the embryo. So how will we compare which embryo is having which band? So where if the band is covering the same distance as covered by HBA, that means that HBA was present. So you can see that HBA is present in 1, it is present in 2, it is present in 4. But the embryo third is lacking HBA because the band is just absent here. Now same goes for HBS. HBS covered this much distance. So we can see if HBS covered this distance, it is present in the second embryo, it is present in the third embryo, it is present in the fourth embryo, but it is not present in the first embryo. So we can just decode the genotype from here, that the first embryo is HBA, the HBA, and the second is HBAS, as the third is HBS, as the fourth is HBAS. So we can just because that's what they will ask us to fill in the next table, so we can do that. State the purpose of using DNA from individuals of homozygous HBA and HBS. These are used as the standards to compare the distance traveled by other fragments, so they can be compared on the basis of the position on the gel. We can mention the genotypes. So further, they are asking discuss the social and ethical consideration of gene testing for embryos for genetic screening. So if now is found, abortion is averted because the couples can who are carriers, they can still have healthy babies by IVF because they can select the embryos. However, the embryos that are not implanted, they are discarded, and this may be conflicting with religious beliefs, and this may not be very ethical to do. So it's having pros and cons both. Now, this question came in Feb March 20, variant 42, and it was a tricky question in which they basically gave the sequence of DNA. So to identify function of a gene, scientists can insert a DNA. We know they are asking you to define recombinant DNA. So recombinant DNA is produced when DNA from 2 sources are mixed together and combined together. This is a plasmid, then the name of plasmid is given to you. The plasmid is having a promoter. There is a multiple cloning site MCS. There is a gene coding for green fluorescence protein. This is used for a screening marker. We know that every recombinant DNA, the bacteria that will take up this plasmid will produce a green fluorescence protein that will fluoresce in the ultraviolet light. So this can be used as a screening tool. This is a gene for antibiotic resistance. Initially, these were used, but now these are not used for screening tool. GFP is added for noting down the screening properties. So they are asking to explain the purpose of including gene of fluorescent protein. Plasmid, the GFP gene produces a protein that fluoresces green in the ultraviolet light. So this GFP gene is used as a marker on this to screen the bacteria that helps in identifying the bacteria that did take up the recombinant plasmid. So what next what they have done. Then next they have given to you MCS, and this information is given to you. So MCS is basically the multiple cloning sites which are shown to you. So we are going into the detail of this multiple cloning site, what actually is present here. So what they have shown to you, I'll just move on to the next page now. So they are saying the gene of with unknown function was inserted into the multiple cloning MCS region. The MCS contain target nucleotide of number of different decision nucleotide into nucleases. The nucleotide sequence of MCS is shown. So figure 5.2 shows to you the MCS region. So this entire sequence is displayed to you. 5' to 3' is the upper strand. One strand is shown to you. 3' to 5', the opposite strand is shown to you. You can see complementary base pairing between all these nucleotides. The nucleotide sequences targeted by 6 excision endonucleases are shown to you and the way in which they cut. So you can see BAMH1 produce a cut in this way, and all these are producing cut end, sticky ends. So except for this SMA1, this enzyme, this will not produce a sticky end because it will produce ends which are blunt. Although all others, they are producing sticky ends. So you can see this from this diagram only, that all the enzymes, they are producing sticky ends, whereas this enzyme will produce blunt ends. So this information is given to you. They are saying that describe a type of end product produced when DNA is cut using restriction endonuclease SMA1. So the end product would lack sticky end, that would be having blunt ends. So this, keeping this in mind, let's just go to the next page now. What they are showing next. Scientists use two electrician endonucleases shown in figure 5.3 to obtain the gene with the unknown function of inserting into the segment. Figure 5.4 shows the gene obtained after cutting with these two restriction endonuclease. So they are using two restriction endonuclease to cut the gene, to cut it at the MCS region to insert the gene. The DNA start codon ATG and the DNA stop codon TA are shaded. So we know that this is the DNA sequence of the gene which they have inserted. The start codon and the stop codon are also shown to you. So this is the start and stop codon are shown to you separately. So basically they are the part of the gene which was inserted. They were initially not present. So when you cut, you need to add the start and stop codon as shown in figure 5.1. Although because that's how the gene will work. Without start and stop codon, it cannot work. They are saying name two restriction nucleases in figure 5.23 that were used to cut MCS of this plasmid. So the gene shown in figure 5.4 could be inserted. So what we need to do, we need to just look at the restriction site of these. So if you look at the restriction site, we know that the restriction site is having the end TCGA at 5 prime. So the enzyme must cut this, and the opposite must be TCGAC 5 prime here. So if we go back and we'll see that the two enzymes which are cell 1 and Zol 1, these are the ones that were producing the cuts at the specific site in figure 5.2 and 5.4. So if we go here in this diagram, we can see that cell 1, this enzyme, it produces a cut in such a way that TCGAC, this line ends up being produced. So this is the site where it is producing the cut. So this is the complementary sequence to it. And similarly, the other enzyme which is producing this cut site, it is present here. So this enzyme produce the cut here. What I have done, I have added annotations here to just point out where this restriction endonuclease will be working. So this restriction endonuclease cut this side, this restriction endonuclease cut this side. So when this cuts, so this is the kind of fragment that will be produced. In the next part, they are asking us to shade the nucleotide sequence that actually got removed. So what are they saying? Furthermore, they are saying to identify the function of the gene, it is important that the gene can be easily inserted to the plasmid. So that's one reason why scientists use different restriction sites at 5 prime and 3 prime end of the gene for inserting the gene into the plasmid. I'll come to figure part 2 later. So why they use different restriction enzyme? Because when they use different restriction enzyme, so the both sticky ends they will not be complementary to each other. So when they're not complementary to each other, they cannot anneal back to form closed plasmid. The closed plasmid ring cannot be formed. How the cut ends can be joined together? They can be joined by DNA ligand. Now, question part 2 was on figure 5.2. Draw around the group of nucleotide in MCS that were removed to insert the gene shown in figure 5.1. Now, this is the gene that they have inserted. If they have inserted this gene, the start and the stop codons are there as well. So you need to find out the sequence 5 prime TCGA and the base pair GC. So if I look for this, what I did, I just marked it here and I started to look. So I found this condition here. This was found here, and basically I had my GC present here as well. So what is this? This is the restriction site, which is where this enzyme is producing a cut. Similarly, at 5 prime end, you have 5 prime TCGA. So this is the cut. This is the point where the cut will be produced. And again, you have a base pair GC here. So what I did on the 5 prime end on the opposite strand, I looked for this. So on 5 prime end, I was able to find TCGA. This was my sequence here. And this base pair GC is there. So this means that both ends, GC and GC, must be there in the sequence, together with a sticky end at this end and a sticky end at this end. Now this is the region that I need to find out on my diagram. So I'll go back to figure and I'll just look. So this is the fragment in the center, and these are the parts which actually are the overlap, you can say the sticky ends. So I hope this was helpful for you. That's it from my side. Thank you so much.